Hello students. Today we are going to start with the next chapter, that is sexual reproduction in angiosperms, or this angiosperms are also called as flowering plants. So as the name indicates, so the plants bear flowers. So and these flowers, as we have studied in the previous chapter, that is one of the reproductive phase, how it is indicated in plants by the production of flowers. So flowers are meant for a sexual reproduction in angiosperms. Okay, so before going to start the actual, as I said earlier, the sexual reproduction always is studied by understanding the three events, that is pre-fertilization event, fertilization event, and post-fertilization event. In the same way, in this chapter also we are studying the sexual reproduction in flowering plants by undertaking such events, okay, under the headings pre-fertilization, fertilization, and post-fertilization. Before starting pre-fertilization, we'll understand the structure of a flower. Typical flower, already we know the structure of typical flower, though once we revise. Okay, yes. The flower, actually the flower consists of 4 whorls. Okay, flower consists of 4 whorls, and these flowers are called as modified shoot. Okay, they are nothing but the shoot which is modified into floral buds. So this happens due to some hormonal changes also. Some hormones like auxins, gibberellins, and florigen hormones are responsible for the production of flowers. And what happens in the shoot, these buds are converted into inflorescence. In such inflorescence there will be formation of flowers. So hence we can call flowers as a modified shoot. Okay, and this flowers also, it is used everywhere, like we give importance culturally, socially, ethically for flowers, right? So these flowers are modified shoot, and they consist of 4 whorls. So those are petals or corolla, sepals or calyx, androecium, that is the male reproductive part of a flower, and gynoecium, a female reproductive part of a flower. So within these two, our petals and sepals are considered as non-essential whorls, whereas gynoecium and androecium considered as essential whorls. Okay, it doesn't mean petals and sepals are useless. Okay, so sepals meant for the protection of the floral bud, and petals are meant for pollination. They attract the insects or any pollinating agent to undergo the pollination. Okay, why it is said essential and non-essential? It is based on, because we are talking about sexual reproduction here. So within the sexual reproduction, so the reproducing parts are the gynoecium and androecium. Hence it is called as essential whorls, and our sepals and petals are called as non-essential whorls. Okay, as the diagram indicates, I have written gynoecium. This gynoecium is commonly called as a pistil or carpel. What it contains? It comprises 3 different parts. Okay, the lower bulged portion where the ovule is incorporated, this portion is called as ovary. The long stalk, okay, tubular stalk, filament like structure, tubular long filament like structure called as style, and above there is a flat surface for the landing of pollen grain. That structure we call it as stigma, togetherly called as pistil, carpel, or gynoecium, and this is the female reproductive part of a flower. Okay, next comes to the androecium, or it is also called as stamen, it consists of two different parts. Okay, the bilobed portion called as anther, and it is attached to a stalk filament, long slender filament called as filament. Togetherly it is called as stamen and on androecium, and all these are placed sometimes within the thalamus, that we have studied based on what we call superior inferior ovary. Yes, the placement. Okay, here like by the diagram we can indicate here the ovary is superior because above the thalamus, so all are arranged on the surface of the thalamus. Okay, and there is a stalk where this flower can be attached to the stem or any leaf inflorescence structure, that is called as pedicel. Understood? And other thing we know, colorful, attractive, varied colored, varied sized petals. This petal are also called as a corolla. And one more, green stalk, which is protective in nature, which is leaf-like structure called as sepal or calyx. Okay, so this is the structure of a flower. Okay, practice this diagram. It is very easier, and try to label as many as possible. Okay. So as already we have discussed, okay, about the basics thing which is takes place in the different events of sexual reproduction by means or with respect to plants also we have studied. Okay, there will be gametogenesis, gamete formation, and those gametes are transferred by the pollination. Fertilization takes place. In angiosperms we know there are special kind of fertilization, what we call double fertilization, triple fusion. Next, after this double fertilization, triple fusion, what happens? There will be post fertilization event. Zygote develops into embryo, endosperm development takes place, seed and fruit development. Okay, this is what we are going to study in this chapter. Okay, we'll start with the pre-fertilization event. So before studying the gametogenesis, we know the pre-fertilization event consists of two sub-events. One is gametogenesis, the second one is gamete transfer. Okay, so before going to the gametogenesis, we should understand the structures of this androecium as well as gynoecium. Okay, what we'll do, we'll study first the structure of this androecium, the ultra structure, and we'll go to the production of the male gamete. Next, similar comes with the gynoecium structure and the female gamete production. Okay, yes, so we'll start with the structure to understand. Okay, the structure of anther, or it is also called as microsporangium. So as we know, these anthers are bilobed. Okay, both the lobes are connected with the help of connective tissue, and this is held by a stalk. We call it as filament. Yes, and these are anther, which is connected by the connective tissue. Okay, the anther, this anther is also called as sometimes diathecus. It is also called as diathecus. Why? Because of, we know there are two lobes. Okay, just imagine there are two lobes. This is made up of two lobes. Each lobe contains like this called as theca. Okay, each lobe contains theca. So hence it is called as dithecous. Okay, and these structures called as microsporangium or microsporangia. So there are total 4 microsporangia at the 4 corners. In turn, later when the development takes place, pollen grain development, this microsporangia itself called as pollen sacs, that is the storage unit of the pollen, developed pollen grains. So this is what the structure. We'll study the correct proper internal structure of anther. Okay, so later we can go through the microsporogenesis. As I said, it is made up of 4 microsporangia at the different corners, and this is our connective tissue where they connect the 2 lobes. Okay, so I'll focus this portion and I'll draw here. Okay, what are the different internal structures of 1 microsporangia? There are remaining 3 microsporangium. Okay, so I'll draw the structure. So this is one highlighted portion of one single microsporangium where there are few layers of cells. It is made up of several layers, and each layers has its own function during microsporogenesis or during the maturation of pollen grain. Okay, so the outermost layer, here you can see this outermost layer. This outermost layer we call it as epidermis. The inner layer, the second layer, is called as endothecium. Okay, this next to the endothecium there are 2 layers. This we call it as middle layers. Okay, so next layer, this layer, call it as tapetum, and these cells, okay, these cells are nothing but our sporogenous cells or completely tissue which contains what? Our pollen mother cell. Okay, or sometimes it is also called as microspore mother cell, PMC or MMC. Okay, and we know this is our connective tissue. Okay, and can you see this blue line where I have indicated there? One more time I'll draw here because it is very important, called as a tapetum layer, which is surrounded towards the microsporangia. Each microsporangia is surrounded by one layer called as tapetum. Inside the microsporangia there will be presence of the sporogenous tissue which contains, which are made up of our pollen mother cell, or it is also called as microspore mother cell. Hence it is called as microsporogenesis. I have told you the production of microspore. Okay, so okay, we'll study the layers of the TS of anther. The outer layer called as epidermis, which in turn helps or functions as a protective layer. Okay, gives protection for the inner layers. Endothecium is the layer which helps in dehiscence. Dehiscence means dispersal. Whenever the pollen grains are get matured and they want to disperse out, okay, from the anther, it should disperse out, right? For that dispersal, this endothecium helps. Endothecium helps in dehiscence of anther, so that anther will open and the pollen grain will be dispersed outside for the pollination process. Okay, next middle layers. So middle layers role is very short. Sometimes protection also, and sometimes giving nourishment, little nourishment during the production of pollen grains. Okay, and after the pollen grain production, they get this middle layer get degenerated completely. That is the fate of our middle layer. Next come to the tapetum. It is very important layer. There are two questions they may ask: the role of endothecium, the role of tapetum. Endothecium is dehiscence of anther for the dispersal of pollen grain. Tapetum is specially nourishment. What is the function? Giving nourishment for the developing pollen grain. Okay, this tapetum layer gives nourishment for the developing pollen grain. Okay, this is what the function of tapetum. And we know connective tissue by the name, and it indicates it connects the two lobes to be intact together. Okay, so this sporangia, as I told, which contains PMC or MMC cells, pollen mother cells or MMC cells, what happens? They undergo the mitotic division and they produce pollen grains within the what? Microsporangia. Later, after, when there is a production, lot of production of pollen grains, this microsporangia called as pollen sacs. Okay, the pollen containing chambers, they are get converted into beginning it was filled with the sporogenous tissue, that is young anther. Okay, how we can differentiate young anther and matured anther? Young anther contains within the microsporangia sporogenous tissue, whereas the mature anthers contains pollen grains within that. So hence those are called as pollen sacs or pollen chambers also. Okay, so this is what the structure of microsporangia or anther. So next we'll go with microsporogenesis.